Well, here's part two of amino acids. And really what I want to do for part two is just go over some aspects of the different types of amino acid chemistry that the side chain groups, and that's mostly what I want to focus on, not the alpha amino or alpha carboxyl group, but just the side chain groups and what kind of chemistry they're capable of doing. And that is a can of sparkling LaCroix sparkling water, natural mango essenced, whatever the heck that means, but mostly I'm thirsty. So periodically I'll have to stop the video to take a sip. I bet you some of you thought that might have been beer or something like that, but it's not. Purely non-alcoholic beverages, although I am old enough to drink, turns out. So it's actually about time I get this out of the way. It turns out that my favorite amino acid is in fact cysteine. And so I'm going to start with cysteine. And cysteine has some interesting reactivity. And this sulfur, this whole group here is called a sulfhydryl or a thiol. So that would be a sulfhydryl, also known as a thiol. And really, if you look at it, it bears a strong resemblance to an alcohol. The only big difference is that there's a sulfur in place of an oxygen right there. And if you remember on the periodic table, sulfur is mighty close to oxygen. It's right below it. So anyway, you might expect them to have similar chemistries. Yeah, they do in some ways have similar chemistries, but there's some differences too. And one of them has to do with this first reaction known as a disulfide formation. Now, a little bit about the nomenclature before I get going. Whenever we deal with an amino acid that is actually part of a protein, so it's a free amino acid, so it's not a free amino acid, so like here, this is a cysteine that's actually part of a protein, so it's lost its free amino group and its free carboxyl groups. We're just really dealing with largely the alpha backbone, we call this part, which I'll talk about later when we get into protein structure, but this is called the alpha backbone, and then the different side chains come off of that alpha backbone. So here's the alpha carbon, here's the beta carbon, and whenever we deal with proteins that are in this kind of form where they're part of the part of the protein, we call them residues rather than amino acids. So if we say a cysteine residue, we're referring to a cysteine that is incorporated into a protein. I could do the same with alanine or glutamate or tyrosine. It's all the same. So whenever you see the term residue, that's just referring to an amino acid that is part of a protein. So this is showing a chemistry that takes place that leads to the sort of joining up of these two sulfhydryls to form something called a disulfide. And this is an oxidation event. And in fact, many times these disulfide sort of formation, where you're back and forth between the disulfide bond and the free thiols, oftentimes that is used in the cell as a way to measure the oxidative stress of the cell. I'll show you in a little bit when I go over glutathione, which is going to be another version of this type of chemistry. So you can see you got half an oxygen, you get formation of water, these two hydrogens, you get water, and you get then this disulfide. And this is a covalent bond, but it is readily reversible in the presence of other sulfurs, so thiols, for instance. So I'll give you an example. If I had another thiol here, and so this could be another cysteine, it could be another small molecule. And that small molecule, if it has a thiol, is capable of interacting with these. So what you need, and I didn't really draw that up here either, but you really need some kind of a base that can abstract that proton. So now it becomes deprotonated. It will have a negative charge. So this does the same thing. It will become deprotonated, have a negative charge, and then this becomes nucleophilic with the negatively charged sulfur, and it attacks one of the sulfurs, and then the other one leaves. So you get kind of a nucleophilic substitution reaction, but dealing with sort of this, what we might call a thiol exchange. And when you have disulfide formed between two cysteines, we just call it a disulfide. But when you have a disulfide made between two different molecules, one might be, say, beta-mercaptoethanol, and the other one could be cysteine, we would refer to that as a mixed disulfide. When you have two cysteine residues that come together and form one of these disulfides, we call it cysteine. Cysteine is what we call two cysteines that are in a disulfide bond interaction. You'll see that later when we go through some primary structure stuff a little bit later. One compound that's often used to make mixed disulfides, and actually it can be used to quantitate how many free sulfhydryl cysteines, reduced cysteines, are present in your sample, in your protein. We can use something called 5,5'-dithiobis 2-nitrobenzoic acid, DTNB. Sometimes it's got another name that you'll see periodically, Ellman's reagent. And the way this works is a sulfur from a cysteine, for instance, gets deprotonated by some kind of base. It could be either OH minus if it's just pure water, or it could be another molecule capable of accepting a proton from the cysteine, so it would deprotonate it, and then a deprotonated cysteine having the negative charge could do that nucleophilic substitution reaction that I mentioned about before, and one half of this becomes a leaving group, so you get something called 5-thiol-2-nitrobenzoic acid. And what's interesting is this guy absorbs at 412 nanometers, so you can actually use UV-vis spectroscopy to visualize, and then use Beer's law. You may remember Beer's law, where you have absorbance is equal to the extinction coefficient, also known as the molar absorptivity, times the path length times the concentration. And I'll actually cover this a little more detail later when we get into primary structure and we get into how do you determine protein concentration, one of the things we'll talk about. But anyway, this is just, you did this back in the olden days in general chemistry, so you may remember it. If you don't, you might want to look it up. Beer's law. And it's a way to determine the concentration of something by measuring its absorbance at a given wavelength, and then using this constant, this proportionality constant, known as a molar absorptivity, or I call it, and a lot of people also call it, an extinction coefficient. Those, I don't know if you can hear those clicks, but that's me adjusting my recliner. It's about 12:30 a.m. as I'm talking to you virtually through my iPad. Anyway, so where this becomes useful is, let's say you have a protein that has a shape and it has a bunch of sulfurs on it. And you want to know how many sulfurs are sticking off the surface of the protein. Well, sometimes that's a useful thing to know for a variety of reasons. If you want to understand, if you wanted to selectively, say, label one of these cysteines and not all of them, you might have to know, well, how many cysteines are available on the surface that could interfere with an experiment. Well, here's one way you can determine that. And you could do that by simply reacting with this Elman's reagent, and then for every cysteine that forms a mixed disulfide, there'll be one of these 5-thio-2-nitrobenzoic acids that pop off in a 1-to-1 ratio, and so you can follow this at 412 nanometers and use the right kind of math, and you can determine the concentration of cysteines that you have. And then you can get a stoichiometry. Do I have 3 cysteines per protein or 1 cysteine per protein? So this is a technique that actually is widely used. I've used it myself in my research. And when I teach Biochem Lab, on occasion, I will, well, every time I've ever taught Biochem Lab, I've actually had people use this process to count how many cysteines that are accessible on the surface of the protein. So cysteine has a lot of other types of chemistry it can do, and I'm going to look a little bit more at disulfide bond formation, but really I'm going to look at the formation of a more reactive species known as a sulfenic acid, and we'll see that that actually plays a big part in disulfide formation. Whoops. But before I jumped the gun, I shouldn't have talked about sulfenic acids yet. First, I mentioned glutathione. Glutathione is an interesting molecule. It's actually a special type of peptide. It has 3 amino acids. And I want you to just look at this glutathione and see if you can recognize the 3 amino acids that are there. Now, don't let this give you any major hints. Glutamyl, cystenyl, glycine. Well, I guess that should have given you the hints. But can you circle them and find where the glutamate is, where the cysteine is, and where the glycine is? And it's a little trickier than you might think. I'll show you. So right here, this would be the glutamyl part, and normally it's this carbonyl that would make a connection to the amino nitrogen to form the peptide bond. But in the case of glutathione, it's actually the carboxyl group, the gamma carboxyl group, that's where the gamma comes in here, of glutamate that makes this peptide bond. And then the next amino acid is, if you look right here, this is cysteine, and the giveaway, of course, is the thiol. And then the last one is glycine. So not all amino acids are actually joined together the same way, and not all of them eventually lead to the formation of a protein. Here's a case where this little special type of tripeptide is made, and it helps regulate the oxidative sort of, not stress, but it helps reduce oxidative stress in a cell, because these guys can gobble up oxygens and form these disulfides. And so that's one of the ways that they can deal with this. And so one of the natural ways that your body fights oxidative stress is uses this molecule glutathione, and it forms a disulfide. And in this case, the disulfide is between 2 cysteines that are found in this tripeptide known as glutathione. So I alluded earlier, a short time ago, to this thing called a sulfenic acid, and what you see here is a sulfenic acid. It's like a sulfur attached to an alcohol group, so it's SOH. And this is a very short-lived, transient, reactive molecule that can go to several fates depending on its environment. So one of the groups that can be formed. Well, let's back up. So how it gets formed is you get this SH gets an one half of an oxygen reacts with one of the SHs, and that's how we get the SOH. So this is called a sulfenic, that's right here, a sulfenic acid. Now the sulfenic acid can go on and do several different things, and here's just a few of them. So in one case you can get two sulfenic acids to react with each other, and you get this formation of a S double bond O. We'd call that a sulfoxide, and that could be in a disulfide linkage. So this looks like a disulfide linkage with a C double bond O present. So that's one of the fates of the sulfenic acid. The sulfenic acid can also go on to be further oxidized. That's what this is showing. So now you get to sulfonic acid and different levels of oxidation. So you can get, I'll show you kind of what this would look like. So this would be SOH, and then this one would be RS. It's going to have, it'll be look like that, and then you would get another possibility would be RS OH. So that's a bunch of different oxidations. So it can react with oxygen further to further go up the stream, or you can get a reaction with a sulfenic acid and a sulfhydryl to form a disulfide. And so in the common sort of pathway for disulfide formation is, in fact, you get the formation of a sulfenic acid and then the sulfhydryl from the nearby sulfhydryl, I should say. You got a base, picks off the hydrogen, then that attacks there, and this OH is released. And that's how we get between, stoichiometrically, we have one proton and one OH that are released, so that is where the water comes from. So you add the two together. So in this way, you can see that the sulfenic acid is a reactive molecule that can be made from cysteine sulf chains, and then cysteine is a pretty good nucleophile. I've already referred to it a few times, and so I want to talk a little bit about some of its other chemistry it can do, some of which happens in the natural course of a chemical reaction, like in an enzyme catalyzing a reaction. In fact, in grad school, I studied a protein called 3-hydroxy-3-methyl-glutaryl coenzyme A synthase. It's a mouthful. That's why we call it HMG-CoA synthase. And in the reaction mechanism, acetyl-CoA, you've probably heard of acetyl-CoA if you've studied anything about glucose metabolism like glycolysis or the Krebs cycle. I find most students have had at least a little exposure to that. We'll get into that in much greater detail later on in this semester. But what acetyl-CoA is an important molecule because it has these, excuse me, I got the hiccups, these acetyl groups. And the way this works is coenzyme A is a really large molecule, and it actually turns out to be a pretty good leaving group. It's very stable by itself when it's not part of, doesn't form this thiol ester group. This is called a thiol ester. So it's like an oxo ester, going back to your organic chemistry days, except just like before, just put an S in place there. So we have thiols and thiol esters. Anyway, going back to what I was saying before, I'm going to clear this out a little bit here. What's happening here is you get base subtraction of a proton, and then the sulfur here can be a nucleophile. It attacks the carbonyl, and then the coenzyme A is kicked off as a leaving group, and now you have a new, if this is a protein, you would have acetylated this protein. Acetylation, where you form a thioester, is a type of acylation. Acylation is whenever you, in the case of a thiol, excuse me, you get some kind of an ester. You can get acylation that leads to an oxoester as well. And when it happens to be a 2-carbon unit, we call it acetylation, because you have an acetyl group transferred. And this can happen in a cell. It can help regulate protein activity. Like when the protein's acetylated, it's turned off or on. And when it's deacetylated, you can reverse the process, whatever you just started. But also it plays a role in chemistry and causing carbon-carbon bond formation and some other types of things like that. Things we will see later on when we get into the glycolysis. So let's move on and see what else we can look at. So cysteine is a nucleophile. You can get something called acylation, which leads to the formation of a thioester. The other type of very similar chemistry, it even sounds the same, but if you look at it carefully, it is different, and the final product is different, is known as alkylation. So you can also alkylate things. Alkylation is a very similar process. This would be an example using something called iodoacetate. And iodoacetate is commonly used in protein chemistry studies because things like sulfhydryls, like this thiol, can act as a nucleophile, attack the acetic acid part, this carbon here. Iodine is a good leaving group, and so it leaves, and so we get this alkylation. Now, acylation forms something, as we just mentioned before. When you acylate something, you get an ester. In this case, you get an ether. So this leads to the formation of a ether. And the thiol in an ether, if you remember, it's a carbon, oxygen, carbon. And the thiol ether would be carbon, sulfur, carbon linkage. So this is what you would get for alkylation. And what's kind of interesting about this, if you look carefully in this particular example, and I find this fascinating anyway, is imagine if you wanted to change the functional group at this cysteine. So let's say I wanted to, look what happens. What amino acid does this now best represent? So we just had a thiol, then when we alkylate it, we just put a carboxyl group on there. And so this kind of makes a long aspartate or glutamate analog. So it's a way that you could do chemical mutagenesis on a protein. You can change the functional group of the amino acid by simply alkylating it, and then you convert it from a thiol to carboxyl. And people do that, and a little later we'll talk. I think I really like this topic, so there's something called chemical rescue, and I'll try to talk about that in class. And then I might bring it up in another lecture as well. Anyway, the point I want you to get from this is cysteine can be a nucleophile. So you're probably going to want to go back and dust off some of the organic chemistry texts you've had and kind of think about what does it mean to be a nucleophile. So then we get another really common and important type of reaction chemistry, and it deals with the 3 amino acids that have OH groups on them: alcohols, serines, threonines, tyrosines. And so we deal in phosphorylation. Phosphorylation is another type, really, of a nucleophilic substitution reaction. So if you look at this, here's ATP, which is the source of the phosphate. So ATP, and the way that I've represented it is I drew ADP, adenosine diphosphate, with the gamma phosphate. This is called the gamma phosphate. And what happens is an alcohol from either serine, threonine, or tyrosine, there'll be a base subtraction of a proton, nucleophilic attack on this phosphoryl center of the gamma phosphate of ATP. ADP is a leaving group, leaves, and you're left with the phosphorylated amino acid, either serine, tyrosine, or threonine. We will actually look at this in greater detail in terms of what are the implications of having your serines, threonines, and tyrosines phosphorylated, and that will come up in the topic that will cover dealing with something called signal transduction. Some of you have probably heard this in other classes, and we will look at it too, but maybe from a little more molecular slant. But I want you to see the chemistry here. So there's not magic here. It's just nucleophilic attacks again. The thing that I don't know if I can't remember which biochemistry class I mentioned this in, but what makes biochemistry kind of cool to me, on top of a lot of things, but one of the things is if you think back to your organic chemistry experience, most of the things you've done were not done in the presence of water. In fact, water is very reactive, and it can really undo a lot of the chemistry that takes place in organic synthesis and making things. So you want to limit the water. A lot of times people will so-called dry their solvents and try to get rid of the water. But in biochemistry, there's a huge amount of organic chemistry going on in the presence of water. And that makes it kind of interesting. And a lot of it's facilitated by enzymes. And so this particular reaction is facilitated by an enzyme type called and we'll learn more about kinases. This is what it's a type of kinase. This is called a protein kinase, is the type of enzyme that catalyze, kinase. So Kinase. It's the type of enzyme that helps facilitate phosphoryl transfer from ATP to either serine, threonine, or tyrosine. I think it's kind of cool. And by the way, when these amino acids are phosphorylated, think about this. When they are phosphorylated, think about how you've changed that side chain. So you see, one of the reasons why the side chains can be modified is it actually adds a little more diversity to the population of amino acids and functional groups that are available in an enzyme or a protein. I keep saying enzyme. Enzymes are proteins. So I want to be a little more generic in proteins in general. So it turns out when you phosphorylate something, look what you're doing. You're adding a phosphate group, and this actually is a phosphoester that you form. It's a phosphoester. And you take an alcohol and convert it to, think about this. This is polar, non-charged alcohol right here. And then you put a formal negative charge on that same spot. So imagine how that might impact something like a protein structure or function. You're taking an alcohol group and replacing it with a charged, bulky group. And what are you replacing? Well, really, technically, you're replacing this hydrogen with this phosphate. Otherwise, this part is the same. So that's a pretty dramatic change. And when you do that to a protein, it can drastically change its structure, its surface contour and charge. It creates new binding sites for other accessory proteins to come now and recognize that place on the protein where it hadn't recognized it before, because it's looking for that phosphate. And so you see that kind of thing happen. Here's another real common reaction. Tyrosines, in addition to being phosphorylated, they do function as nucleophiles, as you're seeing up here. The alcohol can be a good nucleophile as you deprotonate the alcohol. The negatively charged oxygen is reactive. Well, you can do that with tyrosine, is one example. So you can deprotonate the alcohol, and then they get a nucleophilic attack on something that's electrophilic, that has room for the new electrons, and that leads to, here's this example is, this is called, hopefully you recognize it, but you probably, well, I shouldn't say, I say hopefully, maybe you recognize it. This is a type of anhydride. Anhydride, my spelling is not real hot today. Anhydride. Sorry about that. And in particular, it's two acetic acid molecules, so we call this acetic anhydride. Very reactive. It's got good nucleophilic centers around these carbonyls, and so essentially now you get the nucleophilic attack, and this leaving group is acetic acid, or it's acetate, very stable in water, happy to be acetate, and then you get this acetylated. Remember, it's acetylated because you get the formation of an ester, and it's a type of acylation. Acetylation is a type of acylation, so this is acylated. You get the formation of this ester. So tyrosine can do that, and if you remember, in the case of the tyrosine alcohol, it has a pKa of like right around near 10.5 plus or minus 0.2 or something like that. I always forget what it is exactly. But the fact that it has a pKa means that this proton can be removed, and it's acidic. So even without the assistance of another base, when the pH is high enough, in fact, when the pH is above 10.5, when the pH is above the pKa, this group will be deprotonated. That's how that works. So then it becomes a good nucleophile. And then looking further, this tyrosine, in addition to having its reactive alcohol, the fact is it is electrophilic, and tyrosines are pretty susceptible to things like nitration. And one reason why nitration becomes important is these nitrates and these types of molecules, NO2, turns out to be found in pollutants, and these reactive groups can then lead to the nitrated tyrosines, which is oftentimes, like, if smokers would have this happen. Anytime you have other types of combustion products that involve nitrates, your body picks those up as you breathe, and these things can actually be pretty toxic and harmful to a protein, because when you're adding this NO2, now you're changing again the polarity of the aromatic, generally nonpolar ring, and now you're putting a polar group on it. So this becomes one of the mechanisms of toxicity, in one way in which pollutants of various types can impact proteins. Proteins that have tyrosines can be in trouble if you see a lot of nitrates. This one, where you have iodination, uses a similar type of chemistry where you get the aromatic reactivity, and you get iodine attached to the aromatic ring. And I've seen this done in a laboratory setting for like research. So let's say you wanted to add I-125, which is a radioisotope of iodine. You want to attach that to a protein, so maybe you could tag the protein that you can follow it using radioactivity. Well, iodination of tyrosines is one way to do that. And so they use I-125 and intentionally tyrosine, label the tyrosine so that you can, if you're doing certain techniques where you want to detect a certain protein, you label that protein and then you can follow it using a variety of techniques to detect and identify radioisotopes such as I-125. And then as we move along, another, and I'm not going to exhaustively go through all of these reactions. I just want you to kind of get a flavor for the types of reactions that can take place in that. Some amino acids are reactive in ways that maybe you wouldn't have guessed. So in the case of methionine, I think that's maybe one of those, because typically we call methionine a non-polar hydrophobic kind of side chain. Yet sulfur, this is another example of a thiol ether, and it can go through oxidation to form. This would be a sulfoxide. So this would be methionine sulfoxide, and this is sulfone. This can happen naturally in a protein when under oxidative stress conditions. And you can imagine that now you just made a polar molecule the minute you go through these oxidation steps. And then not only have you gone from largely nonpolar to a polar molecule, you've also now introduced bulk in a complete change in the size and shape also of the side chain. So that's one of the types of reactions. Then this one's interesting, the deamination of glutamine or asparagine. Over time, in an aqueous condition, this would be an example of glutamine deamination. So glutamine looks a lot like glutamate, except there's this amine group here. And you can get a nucleophilic, essentially get hydrolysis happening. Water adds across, and you get ammonia and glutamate. So if you think about it, it's another way that a sort of mutation, so to speak, can happen in a protein. Older proteins that have had longer exposures to water at a variety of different pHs can deaminate, and when glutamine or asparagine deaminate, they become a different amino acid. So they get converted between, glutamine goes to glutamate, asparagine goes to aspartate. So this goes to glutamate, this goes to aspartate. And so if you have a lot of that going on, it may be an indication that your protein is old. But that could lead to changes in function, loss of function, just because you're technically changing the amino acid that's at the position. Wherever this glutamine was, now you would replace it with a glutamate. And so that's a naturally occurring thing that could lead to problems. Now I want you to think a little bit about some amino acids on your own. I want you to think about, I'll give you a few hints here. I want you to think about histidine. Histidine has a nucleophilic nitrogen, so that's one hint. So what would a nucleophile do with iodoacetate? This is called diethylpyrocarbonate, but let's not worry about that one. Let's instead worry about, say, the acetic anhydride. That would be a good one. What would happen? How would it react with acetic anhydride? Not sure why I drew this all crooked. How would histidine react here? And it has to do with nucleophilic attack by a nitrogen. Does it have to be protonated or unprotonated? When is it going to be more reactive in each of these cases? When is the histidine imidazole nitrogen going to be a good nucleophile versus not a good nucleophile? So think about that. And then finally, kind of related to side chain chemistry, is those side chains that can accept or supply protons, in other words, be an acid or a base. Can they donate protons and be an acid, or can they accept protons and be a base? And most of the amino acids that have a pKa listed to them can function both as an acid or a base depending on its protonation state. And so that's what I want you to think about. So just quickly, we'll look at aspartate, or just aspartate and glutamate. They both have this side chain with a couple of extra carbons, right? But they have a carboxylic acid functional group. Well, one of these is the acid and one is the conjugate base. And the pKa's for both of them are right around 4. So over which pH range, for instance, will aspartate or glutamate, knowing its pKa, be a base? It'll be a base when the pH is greater than the pKa, because it will be in that state predominantly. And in this, when the pH is less than the pKa, in other words, it's more acidic, this will be the species that's more predominant. And so you kind of go through the side chains and figure out, when are they going to be able to be donors or proton acceptors? Well, it depends. This could be a proton acceptor because it has a free spot for a proton. This one has a proton to give, so it'll be a proton donor. And that's how you have to think about the different amino acids. So if I go through in my head the amino acids that have this capability, you would have cysteine, tyrosine, and then all the charged amino acids are the main ones you should go through. Technically, you could think about threonine and serine as well. They don't list, most tables will not list pKa's, and so in order for those to be deprotonated, you'd have to have the so-called microenvironmental effects happen that I had mentioned about in the last part of amino acids. So I want you to think about these things. That's going to be good for you to work through. And as usual, you got problems or you need a little help with this, make sure you see me during my office hours, or raise the question in class, and if we have time, we can make it something we talk about. Good night, folks.